will be mainly very short summary of the uh, fellowship, senior fellowship what I had from Parkinson UK, and and what kind of research has been done, and I hope it's first explain what we are doing here, not just generally project about PINK1 mutation. Well, just when we talk about disease, any disease, different people understand it differently. I understand that from the terms of patients, it's one, from the doctors, it's slightly different. Well, from scientific point of view, the Parkinson disease is induced by loss of the dopamine secreting neurons from the midbrain region. But again, the mechanism and what is initial trigger for in each case, still unclear. And what was a real burst in understanding the mechanism was a discovery of the familiar form of Parkinson disease. Explaining why it's important, for example, for other research, because before people use some model which is toxic, and many of these models are just use some kind of pesticides or inhibitors, and these inhibitors mostly affect mitochondria, organella, which we just mentioned several times. And what is important, that all these genes are not all of them, they're predominantly mitochondrial, suggesting that we have something with energy deprivation, or at least we have a problem with every function which connects to mitochondria. And when this project been started, it was almost nothing known about PINK, and almost about all those genes in terms of function, because the genes been found, but what exactly causes any mutation in this gene, or what kind of pathology may be associated with this. And and actually it was a goal of all this fellowship. And on the first stage, we just tried to understand, because the PINK1 was discovered here in this institute, and first what we tried to understand, it was the physiological role of the PINK1 in the cells, and exactly in neurons. And we use one of the most modern technique is imaging. This is one of the indicators which we used in live cells. If I just click, you will see, we can do the 3D images here, but it's not just a picture of the mitochondria in human neurons which derived from the stem cells. It's important information, because the intensity of this indicator tell us what is the value of one of the major characteristics of the mitochondrial physiology and mitochondrial health is the mitochondrial membrane potential, which used by mitochondria to produce ATP, our main energy molecule. And we can see here that PINK1 deficiency leads to the significant drop in mitochondrial membrane potential in all models. It's not something which are related to one of the models. It's we found that in human neurons, as I said, which been derived from the stem cells. It was in some cellular models, which was just genetically modified, or it was found in transgenic animals. What is important? It came when we expressed PINK1 back to these cells, the mitochondrial membrane potential was restored. Okay, we definitely have mitochondrial problem and mitochondrial dysfunction in the cells, and using the very, I mean, the number of approaches, well, this is, I especially put this here because, well, it looks very complicating, but, well, and it's all what we did before. I mean, just trying to understand the mechanism, well, and what is initial trigger for cell death. And we found that pathology in PINK1 is linked to the two major factors. Well, it's energy deprivation is due to inhibition of the transport of the glucose on the plasma membrane, and because of abnormal calcium signaling. Well, calcium signaling is a major ion, which, I mean, well, and sorry, cation, which is produce all signaling between cells and especially between neurons. And when we have this abnormality, it's lead to the cell death, well, and loss of the cells. And if in all body, the loss of the cells, well, we're losing a lot of cells every day, well, for example, skin, well, it's essential for us, but, well, if it's induced cell death in neurons, well, it's it lead to the problems, well, with, and this is a publication which have been, well, what, this paper which we published on the basis of this research, and recently I checked, and it's more than 170 citations since then, which suggest that, well, this paper being attract attention a lot of scientists, and and it's already taken as a basic principle for neurodegeneration in PINK. But it's knowing the role of the PINK in physiology was not just enough. Well, because we know that we have variety of the cells in our body, but why this mutation or deficiency lead to the neurodegeneration of the specific brain area and specific neurons in beginning? And we suggest that it might be because, well, we have a dopamine secreting cells, and it might be excessive amount of the dopamine in that area, which might act as a trigger as well. And but we've been surprised that the mechanism of the dopamine-induced cell death never been studied, and even more, well, calcium signaling was not studied properly. And this is well, what happened with the brain area. Well, this is different cells. It's neurons and astrocytes. Astrocytes is glial cells which